The next speaker is Dr. Smaali from Tunisia and University of Carthage, National Institute of Applied Science and Technology. This is a talk about implementation of biotechnological concept for valorization of Tunisian date waste. So welcome, Dr. Isam.
So first of all, I would thank the organization committee that gave me this opportunity to share with you this presentation entitled Implementation of Biotechnological Concept for Valorization of Tunisian Date Wastes. So this work was carried out in the National Institute of Applied Sciences at Tunis, which belongs to the University of Carthage. As you know, date palm production takes a specific place in the North Africa and Middle East. So among all these aspects and advantages, we can cite the economic development. One pillar of the economic development is the circular bioeconomy, which perfectly suits the valorization of dates co-products via biorefinery approaches to produce bio-based products applied in different sectors. Tunisia is producer of date fruit with an average of 245,000 tons per year, where the major cultivar is Deglet Nour that represents 75%. In terms of export, 42% of these dates are exported to the European country and some African country. Given this production, there is a sustainable loss in this fruit. This from the two stages of the economic chain value, notably in the production and in the sorting when we pour harvest handling. So this date wastes constitutes a renewable feedstock for biorefinery and bio-based products. Their use is limited to animal feed, and our objective is to give biotechnological processes to give new and innovative products. So the concept, what we do in the lab, our concept will take as starting material these wastes without pits and after grinding. One major work was dedicated to the hydrogen production by dark fermentation. This is an innovative approach that was assayed in our lab, but we have developed other road for valorization of these co-products. Is the hot water extraction to produce filtrate of soluble sugars and crude fibers. So for the filtrate, they can be evaporated to give date syrup, and we push the valorization to the food domains to substitute sucrose in sponge cakes as model food, to produce FOS or fructooligosaccharides That's for the date syrup. Concerning the crude fibers, we can either go to the energy sector or in the food sector. So what we propose here is a hybrid approach that permit to develop many products. In this presentation, I will focus in the biohydrogen. And briefly some application in the food sectors. For the fermentative biohydrogen, we start by determining the composition of date flesh. And as you can see, they are good substrated for microbial conversion. We can see here good value for total solids as percentage of wet weight and VS or volatile solids. So these value makes them as good substrates for microbial conversion. Dark fermentation uses anaerobic bacteria which freely and efficiently produce hydrogen with no need of light and at low operation costs compared to photo production. The microorganisms that are involved in dark fermentation are facultative or obligate anaerobic bacteria that produce hydrogen through two pathways, the acetate one and the butyrate pathway. In a typical experiment of biohydrogen synthesis by dark fermentation, we have the biomass, which can be treated or chemically hydrolyzed. Then we should put the inoculum, which can be a consortia of microorganisms or one strain. The main batch here take few days to produce hydrogen and organic acids. So this model was applied to our biomass, and for this purpose, we simplified this process. So as starting material, we used discarded Deglet Nour co-products. For the pre-treatment, we only remove pits and largely milled and rinsed with water. We doesn't add any microorganism to the batch fermenter. The output of this experiment are what we monitor is the biohydrogen via BHP tests with GC-TCD. It's a cumulative hydrogen produced. We studied the microbial community by DNA extraction, by PCR amplification and sequencing, and blasting with bioinformatic tools. And finally, the final metabolites with HPLC. So the results for cumulative biohydrogen, as you can see, we obtain a very interesting value of 222 milliliter of hydrogen per gram volatile solid. It can express like 2.47 mole of hydrogen per mole of equivalent exhaust. This is an interesting value compared to the bibliographic data and assayed for other substrates. So the second part was the microbial community analysis, and our results showed that there is enteric bacteria at around 20% and uninspected lactobacillus at a high value of 39%, and Clostridium genus. It's a typical microbacteria in this kind of bioconversion. So this uninspected lactobacillus works via lactate pathway. And we didn't find here, when analyzing the final products in the liquid after dark fermentation, there is no lactate. So the hypothesis what was verified is that the lactobacillus produce lactate, and this lactate was metabolized by the Clostridium to produce the butyrate. So there is a production of hydrogen via the butyrate pathway, but also with the acetate pathway. So this interesting value, without adding any external microorganism, can be explained by this synergy between these two bacterial genus. I will move now briefly for the second application concerning the food application, and as I said, we worked on date syrup. So date syrup should be different from date rub or molasses, which is brown and dark. So the technique of heating is very important to obtain this date syrup. So in our country we have a problem of sucrose, and there is an active research to substitute sucrose in industrial foods. So the date syrup here was taken as an alternative to sucrose in food products. This is substitution is sustainable and can give a lot of benefit. So we carried out a little characterization of our date syrup. As it can see, there is it's rich in potassium, magnesium, total phenolic compounds. So it's a typical composition that confirm the nutritional quality of this syrup. So to push this experiment and to give final valorization, we perform substitution of sucrose in the recipe of sponge cake. And as you can see here, donc we carried out 4 experiments. We take sucrose as a control, and 3 substitution at 50%, 75%, and fully substituted date syrup. So we here for the control and the last experiment, we have the same bricks of 50, which means 50 gram per 100 gram of material. So this sponge cake are taken to as a model because the recipe was simple and straightforward to realize. So when performing texture properties, as you can see, the fully substituted with date syrup, the SPC3 experiment, showed different properties when compared to the other. It's very clear that all the texture properties are different. Another advantage of this substitution is after cooking, we analyzed the antioxidant activity after cooking, and we found for this assay that we have a considerable antioxidant activity compared to the control with sucrose that don't own any activity. Finally, for the sensory evaluation, the developed sponge cake with date syrup was a good product, as you can see here, for the odor, the taste, the texture, the global appreciation. are very acceptable. The color was less acceptable. It takes, like you see here, it takes a number of 4, rating 4 of 5. The last part is in this food application is the synthesis by transglycosylation of fructooligosaccharides as prebiotics. As you know, prebiotics have a good benefit to the gastrointestinal tractus, and they are used to formulate functional foods. They are innovative products demanded in the market and are very important for the health. So we take beta-fructoofuranozidase, fungal beta-fructoofuranozidase as enzyme, and we carried out a transglycosylation reaction. This is a typical reaction for glycoside hydrolysis to synthesize, to transfer fructosyl moiety to an hydroxylated glycone in the medium. So from date syrup, we synthesizes fructooligosaccharides. This was confirmed and analyzed by TLC, preparative TLC, by FTR, by Malditov, by NMR study, and we demonstrated that the first synthesis corresponds to 1 kistose, a major prebiotics, and you can see the chemical formula. In conclusion, so we used biotechnology process were used to valorize date wastes and byproducts with sustainable way with zero effluents. A multi-objective approach that combined foods and bioenergy was implemented. Dark fermentation was used to produce biohydrogen from discarded Deglet Nour dates with a high yield. It corresponds to an innovative strategy without specific pretreatment or inoculum addition. Endogenous bacteria are sufficient and efficient to convert flesh dates to hydrogen. For foods, date syrup was produced at a pilot scale with straightforward process. It was demonstrated that it can perfectly substitute sucrose in industrial foods. And finally, 1 kistose as prebiotic fructooligosaccharide was successfully synthesized from date syrup by transglycosylation activity. And thank you for your attention.
Thank you, Dr. Assam. And the time for questions.
Thank you for this presentation, which is very clear. So my question is about the organic part. So during producing ethanol, we extract a serum. This serum is very, it's not concentrate. So we know that is the scale up from a laboratory scale to the industrial scale. We need to concentrate that serum into small amounts in order to have a good storage. So have you any idea about how we can make this concentration proceeding? Because we know that is this product is very interesting for agriculture field, exactly for producing fertilizer.
Thank you for this question. Obviously, the term, the word syrup should be used after evaporation. What we extract is just juice or sugar extract. So to speak about syrup, we should evaporate. And the evaporation, there is many techniques of evaporation by heating. So we can evaporate under vacuum with vapor of water, and it can give good results.
Thank you for your response.
Thank you for this great presentation. I have a question regarding the shelf life of the date syrup. And what is the recommended bricks concentrations that you recommend that will keep it? We can store the date syrup because in the market now it is about around from 70 to 80 bricks. This is what in the field.
This is the right value because you should evaporate to obtain 70 to 80 bricks to avoid the proliferation of microorganisms and to conserve it in
And
fridge.
what is the shelf life?
The shelf, really we don't determine the shelf life, but if it is put in fridge or at 4 degrees Celsius, maybe for 6 months.
No, I'm talking about industrial things. If I have tons of date syrups, I'm gonna utilize it for industries. And what is if I can if I you know that if the bricks is more than 80%, there is no bacteria that's gonna be
Yes.
available there. So what is the why I have to keep it in the fridge? If it's 80% bricks, what is the shelf life and the room temperature?
For industrial application, it's a consensus between the evaporation needed, so the electricity consumption, the heating, and the final product. So to get the 80%, we need a strong evaporator to treat huge quantities.
No, this is not my question.
Ah.
My question that I already have date syrup with 80 bricks.
Yes.
I'm gonna keep it in room temperature. What is the shelf life for it?
We don't determine the shelf life. I can't respond to this question, but what I know, when we put it in 4 degree in the fridge or in frigorific room, the 6 months are guaranteed.
Okay. Thank you.
Any more question?
Thank you, Dr. Asa. Actually, regarding the fermentation of the byproduct of this, either you go for biogas or you go for hydrogen. In the case of hydrogen, is there any byproduct in the form of gases or liquids or solids? And which one you prefer, to go for biogas as methane or you go for hydrogen as a source of energy?
We don't prefer, since we don't add microorganisms, so the reaction, this fermentation is spontaneous. So when we do it for the first time and we analyze the gas composition, we understand if the pathway go to the hydrogen or to the methane. So for this case, preliminary assays give us a lot of hydrogen compared to methane. So we push this experiment to produce biohydrogen without adding a microorganism. But if you add an inoculum, you can go over the biogas pathway.
Thank you. Any questions? Okay, thank you,
Thank you
Doctor.
very much.
And your certificate?
Thank you.
Thank you. Is Dr. Muhammad Ali here? I think Muhammad Jawid. Yes. Okay, so we have closed the session and we have wish you a great time. Thank you. Speaker.